Welcome to our deep dive,
Yeah.
into the fascinating world of modern drug discovery techniques.
Oh, yeah.
You've given us a collection of really intriguing factoids about cutting-edge drug discovery techniques, and I can already tell this is going to be a wild ride.
Absolutely.
Get ready to explore AI-designed molecules, gene editing that could rewrite inheritance, and even enzymes built from scratch.
It's definitely an exciting time to be in the world of medicine.
It is.
Some of these advancements seem like they're straight out of science fiction.
I know.
Really?
Okay, let's unpack this first factoid, which has already blown my mind a little bit.
Okay.
Deep learning in chemistry.
Yeah.
Scientists are using AI, specifically deep learning, to design new drug molecules.
Right.
It's like having a digital chemist that can analyze millions of possibilities in seconds.
It's pretty remarkable.
Yeah.
Instead of relying on traditional lab techniques, which could take months or even years, these algorithms can sift through massive data sets of chemical structures and properties to predict which molecules might be effective against a specific disease.
So instead of endless trial and error, researchers can now get a head start with AI pointing them towards the most promising drug candidates.
Exactly.
Wow.
This speeds up the drug discovery process significantly and lets researchers focus on the molecules that are most likely to succeed.
Yeah.
This is a game changer for complex diseases like cancer or Alzheimer's, where finding effective treatments has been incredibly challenging.
Absolutely. What I find fascinating is how these deep learning algorithms actually work.
Yeah.
It's like they've learned to speak the language of chemistry.
Right. They are trained on enormous data sets of molecules and their properties, learning to recognize patterns and relationships that humans might miss.
Yeah.
They can then use this knowledge to predict how new molecules will behave, including how well they might bind to a specific target in the body.
Binding affinity.
Yes.
That's a crucial concept in drug development, right?
Yeah.
It's all about how strongly a drug molecule attaches to its target, which influences how effective the drug will be.
Precisely, deep learning is allowing us to design molecules with much higher binding affinities, which means more effective therapies with potentially fewer side effects.
That's incredible. Now, our next factoid takes us into the world of gene editing with CRISPR, but it goes beyond just tweaking genes.
Oh.
We're talking about CRISPR gene drives that can rewrite genetic inheritance itself.
This is where things get really interesting.
I know.
And potentially a little bit controversial.
Yeah?
A gene drive is a genetic element that can spread rapidly through a population, ensuring that a desired trait is passed on to almost all offspring.
So you're telling me that we could potentially use this to control populations of disease-carrying insects like mosquitoes?
That's one of the most talked about applications. Imagine a gene drive that makes mosquitoes resistant to malaria, effectively eliminating the disease from entire regions.
Wow, it sounds almost too good to be true.
It does.
But how does a gene drive actually work on a molecular level?
It's all thanks to CRISPR's ability to cut and paste DNA. A gene drive is essentially a self-copying genetic element that includes the CRISPR system and the desired gene.
Okay.
When a gene drive is introduced into an organism, it cuts the DNA at a specific location and inserts itself along with the desired gene.
Got it.
Then, when that organism reproduces the gene drive, copies itself into the other chromosome, ensuring it's passed on to the next generation.
So it's like a chain reaction that spreads the desired trait through the population. Fascinating,
Oh,
but definitely raises a lot of questions about the ecological impact of altering entire species.
absolutely.
Let's move on to our next factoid, which takes us from manipulating genes back to the world of chemistry with something called chemoenzymatic synthesis.
Okay, this technique is like a hybrid approach to building drug molecules, combining the flexibility of traditional chemical synthesis with the precision of enzymes.
Okay, break that down for me. We're talking about building molecules, right?
Yeah.
How do enzymes fit into that?
Well, think of it this way. Scientists use chemical reactions to build a basic molecular framework, kind of like a rough draft of the molecule they want to create.
Okay.
Then they bring in enzymes to refine the structure, adding specific functional groups or creating very precise 3D shapes, which are critical for drug effectiveness.
So the enzymes act as highly specialized tools that add the finishing touches to the molecule.
Exactly. Enzymes are incredibly good at catalyzing specific chemical reactions with high precision, which is something that traditional chemical synthesis often struggles with.
Gotcha. This brings to mind the concept of chirality in molecules,
Yeah,
where two molecules can have the same atoms but be arranged differently in space like mirror images.
you're right. Chirality is incredibly important in drug development because the two mirror images of a molecule can have drastically different effects in the body.
Yeah,
One might be therapeutic while the other is toxic.
and enzymes can help control chirality, ensuring that you're producing the right form of the drug molecule.
Exactly. Chemoenzymatic synthesis allows for much finer control over the final structure of the drug molecule, ensuring that it's not only effective but also safe.
That's amazing. So we've gone from AI designing molecules to gene editing, and now hybrid approaches to building them. What's next on our journey through the future of drug discovery?
Let's delve into the world of directed evolution, a technique that allows scientists to accelerate the evolution of enzymes with desired properties.
So instead of waiting for nature to do its thing, scientists are speeding up the evolutionary process in the lab.
You got it. It's a fascinating process that mimics natural selection, but on a much faster timescale.
Okay, I'm intrigued. How do they actually speed up evolution in a test tube? I imagine it's not as simple as hitting fast forward.
It's pretty ingenious. They start with a gene that codes for a specific enzyme and introduce random mutations into that gene.
Okay.
These mutations change the enzyme's structure and function. Then they screen these mutated enzymes to see which ones perform best in a particular task, such as breaking down a harmful protein or catalyzing a reaction needed for drug synthesis.
So they're creating a whole library of mutated enzymes and then selecting the ones that are best suited for the job.
Exactly. It's a bit like breeding super enzymes for drug discovery, and the beauty of this approach is that it doesn't require a deep understanding of the enzyme's structure or function.
Yeah,
It's all about selecting for the desired outcome.
that's a fascinating concept. So scientists can essentially custom evolve enzymes to perform specific tasks in drug development.
Amazing. Now, our final factoid for this part of our deep dive takes this concept a step further with something called artificial enzymes.
This is where we move beyond mimicking natural enzymes to actually designing and building them from scratch.
Wow.
It's a revolutionary field with immense potential.
Building enzymes from scratch, it sounds like we're entering the realm of science fiction. Are we talking about completely artificial molecules that act like enzymes? Yes. These artificial enzymes, or catalysts beyond nature, as some call them, can be designed with specific properties and activities not found in nature,
Okay.
opening up new avenues for drug development and beyond.
So we're talking about creating custom-designed enzymes that can perform tasks that natural enzymes can't.
Right.
What are some examples of how this could be applied to drug discovery?
Imagine an artificial enzyme that can break down a disease-causing protein with incredible precision, something that a natural enzyme might not be able to do, or an enzyme that can catalyze a reaction needed to synthesize a complex drug molecule that's currently impossible to make with traditional methods.
It's like we're entering a whole new era of enzyme engineering. This is pretty mind-blowing.
It is. The possibilities are really endless.
Well, I think we've covered some truly groundbreaking territory in this first part of our deep dive.
Yes.
We've gone from AI-designed molecules to gene drives to custom-built enzymes. It's clear that the future of drug discovery is going to be incredible, and we've only just scratched the surface.
Right, and we still have so much more to uncover.
Before we move on to some real-world examples of how these techniques are being used in part 2 of our deep dive, I want to hear what stands out to you the most. What has you particularly excited about all of this?
What fascinates me is how all of these techniques are pushing the boundaries of what we thought was possible in medicine.
Yeah.
We're no longer limited by what nature provides, but can actually design and build the tools we need to fight disease at a molecular level.
I totally agree. And for me, it's the incredible speed of advancements. It feels like every day there's a new breakthrough, a new possibility. It's an exciting time to be alive and witnessing this revolution in drug discovery.
Absolutely. I'm ready to dive into some real-world applications.
Me too. Let's continue our journey through the future of medicine in part 2.
Welcome back to our deep dive into modern drug discovery techniques.
I'm ready to hear about those real-world examples you promised.
Right.
In the last part, we discussed a lot of cutting-edge techniques, from AI designing molecules to gene drives and even building enzymes from scratch.
We certainly covered a lot of ground, and now it's time to see how these techniques are being applied to develop actual drugs and therapies.
Let's start with deep learning.
Okay.
You mentioned how AI is being used to design molecules, and it seems like something straight out of a sci-fi movie.
It does, doesn't it? But it's very real, and it's already having a significant impact. One striking example is the development of a potential new drug for idiopathic pulmonary fibrosis, a devastating lung disease.
I have to admit, I'm not familiar with that particular disease? What
Ye-
can you tell me about it?
It's a rare condition where lung tissue becomes scarred and stiff, making it incredibly difficult to breathe.
Oh, wow.
There are some treatments available, but they're not very effective, and they come with a host of side effects. Researchers have been struggling for years to find better options, and that's where deep learning comes in.
So how did AI help in this case? Did it design the entire drug molecule from scratch?
Not exactly. Researchers used deep learning algorithms to analyze a massive data set of chemical compounds and identify a molecule that showed promise in preclinical studies. It wasn't a completely new molecule, but one that had never been considered for this particular disease.
So the AI pointed them in the right direction, and they were able to take it from there.
That's right. The AI identified a molecule that had specific properties that made it a good candidate for targeting the underlying mechanisms of the disease from there. Researchers were able to conduct further experiments and optimize the molecule for efficacy and safety.
And what's really remarkable is that this molecule has a completely novel mechanism of action,
Yeah.
meaning it works in a way that no existing drug does.
Mm.
So it's not just about repurposing existing drugs, but discovering completely new ways to treat disease. That's amazing.
It really highlights the power of AI to uncover hidden therapeutic possibilities that humans might miss. Now let's move on to CRISPR gene drives, which I know we both found fascinating
Yeah.
and maybe a little bit unsettling.
Uh-huh.
Are there any examples of these being used outside the lab?
That's what I'm really curious about. We talked about the potential to control mosquito populations and combat malaria, but are there any real-world projects happening right now?
It's still early days for CRISPR gene drives, but there have been some promising field trials. One notable example involves using gene drives to combat malaria in mosquitoes in Africa.
So they're actually releasing genetically modified mosquitoes into the wild.
Yes, but it's not as simple as just releasing a bunch of mosquitoes and hoping for the best. There are very strict safety protocols and containment strategies in place to ensure that the gene drive doesn't spread beyond the target population.
I can imagine there's a lot of concern about the potential ecological impact of releasing gene drives into the environment.
Absolutely. Scientists are proceeding with extreme caution, conducting small-scale trials in controlled environments to assess the effectiveness and safety of the technology. It's crucial to ensure that the benefits outweigh the risks.
It sounds like a delicate balancing act, but the potential to eradicate malaria is huge. Now, let's shift gears and talk about chemoenzymatic synthesis. I'm curious to hear about how this technique is being used to develop new drugs in the real world.
One area where chemoenzymatic synthesis has really shown is in the development of antiviral drugs, which is incredibly relevant given the global pandemic we've all experienced.
Oh, absolutely. Antiviral drugs are definitely top of mind these days. Can you give me a specific example of how this technique is being used?
One example is the production of the antiviral drug oseltamivir, also known as Tamiflu, which is commonly used to treat influenza.
I've definitely heard of Tamiflu. It's been around for a while, hasn't it?
It has, and it's a prime example of how chemoenzymatic synthesis can streamline and improve drug production. The key step in synthesizing oseltamivir involves using an enzyme to add a specific chemical group to a precursor molecule. This enzymatic reaction is incredibly efficient and selective, producing high yields of the desired product.
So it makes the manufacturing process more efficient and potentially less expensive.
Exactly, and because enzymes are so specific in the reactions they catalyze,
Right.
it also reduces the formation of unwanted byproducts, making the drug safer and more effective.
It's amazing how nature has already solved so many of the problems we're trying to tackle in medicine. We just have to learn how to harness that power.
Couldn't agree more. Nature is a brilliant chemist, and we're constantly discovering new ways to exploit its ingenuity for the benefit of human health.
Now let's talk about directed evolution, which you described as speeding up evolution in the lab. I'm eager to hear how that's being applied to real-world drug discovery challenges.
One remarkable example involves the development of a new class of antibiotics, which is crucial given the growing threat of antibiotic resistance.
Antibiotic resistance is a huge problem, and finding new antibiotics is a constant race against evolving bacteria.
It is, and directed evolution is providing us with a powerful tool to combat this threat. Scientists have used directed evolution to create enzymes that can break down the cell walls of bacteria that are resistant to existing antibiotics.
So they're essentially evolving enzymes that can overcome antibiotic resistance.
They start with an enzyme that shows some activity against the target bacteria, and then they use directed evolution to introduce mutations and select for variants that are even more effective. It's a brilliant way to stay ahead of the evolutionary arms race.
It's like we're fighting fire with fire, using evolution to create the weapons we need to fight drug-resistant bacteria. That's really encouraging.
It's a testament to human ingenuity, wouldn't you say? We're constantly finding new ways to outsmart nature, even when it throws us curveballs like antibiotic resistance.
Absolutely.
Yeah.
Now, for our final real-world example, let's talk about artificial enzymes. I'm really excited to hear about how these custom-built catalysts are being used to tackle diseases.
One area where artificial enzymes show great promise is in the treatment of cancer, which, as you know, is a complex and challenging disease.
It's a disease that affects so many people, and finding new and more effective treatments is an ongoing battle.
Scientists have designed artificial enzymes that can target and destroy cancer cells with incredible precision, leaving healthy cells unharmed.
That sounds like a game changer. What makes these artificial enzymes so precise?
They can be engineered to recognize specific markers that are only present on cancer cells, so they act like guided missiles, delivering their payload directly to the target. This minimizes side effects and improves the effectiveness of treatment.
So it's not just about killing cancer cells, but doing so in a way that spares healthy tissue and improves the patient's quality of life.
Exactly, and artificial enzymes can also be designed to overcome drug resistance, which is a major problem in cancer treatment, by creating enzymes that can bypass the mechanisms of resistance.
Yeah.
We can develop therapies that are more durable and less likely to fail.
It's amazing to think that we can design enzymes that can do things that natural enzymes can't. It's like we're rewriting the rules of biology.
In a way, we're no longer limited by the constraints of natural evolution, but can actually design and build the tools we need to tackle some of the most challenging diseases.
These real-world examples really highlight the incredible progress that's being made in drug discovery. It's clear that these techniques are not just pie-in-the-sky ideas, but are actually leading to tangible benefits for patients. It's really inspiring.
It is, and it's just the beginning. I can't wait to see what breakthroughs the future holds.
Me too. But before we move on to our final part, I have to ask, what are the biggest challenges facing these new techniques? It all sounds incredibly promising, but surely there are hurdles to overcome.
That's a great question, and you're right, there are always challenges. One, the biggest, is scalability.
Yeah.
Some of these techniques, like chemoenzymatic synthesis and artificial enzyme production, can be complex and expensive to scale up for mass production. It's one thing to create a new drug in a lab, but it's another thing entirely to manufacture it on a scale that can meet global demand.
So it's a matter of bridging the gap between scientific discovery
Yeah.
and commercial viability.
Exactly. Another challenge is regulation. As we develop these powerful new tools we need to ensure they're used responsibly and ethically, regulation needs to keep pace with scientific innovation to ensure that these technologies are safe and effective for patients.
So it's a balancing act between pushing the boundaries of science while making sure we do so responsibly.
Precisely. And then there's always the risk of unforeseen consequences. We're venturing into uncharted territory with some of these technologies, and it's impossible to predict with absolute certainty how they will behave in the long term. We need to be vigilant and prepared to adapt as we learn more.
It sounds like we need to be mindful of both the incredible potential and the potential pitfalls of these advancements. But overall, it seems like the future of medicine is very bright, wouldn't you say?
I wholeheartedly agree. We're living in an era of unprecedented scientific progress, and the future of medicine is filled with possibilities. Who knows what breakthroughs await us in the years to come?
Now, I'm really curious to hear what you've got in store for us in part 3 of our deep dive. You promised some even more mind-blowing facts about the future of medicine.
Get ready for a journey into the world of polypharmacology and cryo-electron microscopy. Trust me, you won't be disappointed.
I can't wait to see you in part 3. Welcome back to the final part of our deep dive into the future of drug discovery.
I'm excited to delve into these last two cutting-edge concepts I mentioned.
Me too. You left us hanging with talk of polypharmacology and cryo-electron microscopy.
Right?
And I have to admit, those terms sound pretty intimidating.
They might sound complex,
Yeah.
but the ideas behind them are quite elegant. Let's start with polypharmacology. It's a bit of a mouthful,
It is.
but it's a really exciting concept in drug discovery.
Okay, break it down for me. What is polypharmacology?
It's the idea that one drug can interact with multiple targets in the body, kind of like a master key that unlocks several doors.
So instead of just targeting one specific protein or pathway, one drug could tackle multiple disease processes at once.
Exactly. This is particularly relevant for complex diseases like cancer or Alzheimer's, which involve multiple interconnected pathways and mechanisms.
That makes a lot of sense. Those diseases are so complex, it seems like hitting multiple targets at once would be much more effective than just focusing on one.
Right, and polypharmacology also has the potential to simplify treatment regimens. Instead of needing a cocktail of drugs with all their potential side effects and interactions, we could have one drug that does the job of many.
So less pills to take and potentially fewer side effects. That sounds like a win-win for patients.
It does, and it's a more holistic approach to treating complex diseases. Instead of just treating the symptoms, we can target the underlying networks of interactions that contribute to the disease.
It's like tackling the root of the problem instead of just pruning the branches.
Yeah.
I, that analogy. Are there any drugs being developed using this approach?
Yes, there are several in the pipeline. For example, researchers are developing drugs that target both the amyloid plaques and the tau tangles found in the brains of Alzheimer's patients.
So one drug to address two key hallmarks of the disease. That's incredible. Are there any challenges in developing these multi-target drugs? It seems like it would be incredibly complex to design a molecule that can interact with multiple targets effectively.
It definitely presents a challenge. Identifying the right targets and designing a molecule that can bind to them effectively is a huge hurdle. There's also the issue of predicting and managing potential off-target effects.
Yeah.
Even though a drug is designed to hit specific targets, there's always a chance it could interact with other molecules in the body, causing unintended consequences.
So it's a balancing act between hitting the right targets while minimizing the risk of hitting the wrong ones.
Precisely, but with careful design, sophisticated screening techniques, and rigorous testing, researchers are making great strides in developing safe and effective multi-target drugs.
It sounds like polypharmacology has the potential to revolutionize how we treat complex diseases. Now let's move on to cryo-electron microscopy, or cryo-EM.
Okay.
What is this technique all about?
Cryo-EM is a revolutionary imaging technique that allows scientists to visualize the structures of biological molecules, like proteins and enzymes, in incredible detail.
We've talked a lot about the importance of the 3D structure of molecules, especially in drug development. So is cryo-EM giving us a much clearer picture of what these molecules actually look like?
You got to imagine being able to see the intricate details of a protein, its folds and crevices, the precise arrangement of its atoms. That's what cryo-EM allows us to do.
And why is that so important for drug discovery?
Because understanding the 3D structure of a drug target is crucial for designing drugs that can interact with it effectively. It's like having a blueprint of the lock you're trying to open.
So with cryo-EM, scientists can see exactly how a drug molecule fits into its target, which allows them to design better drugs with fewer side effects.
Exactly. Cryo-EM has revolutionized structural biology and is rapidly becoming an indispensable tool in drug discovery. It's allowing scientists to develop drugs with unprecedented precision and speed.
It sounds like a game changer. Are there any specific examples of how cryo-EM is being used to develop new drugs?
One area where cryo-EM is having a major impact is in the development of antiviral drugs. For example, cryo-EM has been used to determine the structures of key viral proteins, including those of HIV and influenza.
And by understanding the structure of those viral proteins, scientists can design drugs that target them more effectively.
That's right, they can design drugs that fit perfectly into the active sites of these proteins, blocking their function and inhibiting viral replication.
It's incredible how technology is accelerating scientific progress. What are some other areas where cryo-EM is making an impact?
Cryo-EM is also being used to develop drugs for a wide range of other diseases, including cancer, Alzheimer's, and autoimmune disorders. It's helping us understand how these diseases work at a molecular level and design drugs that can target the underlying mechanisms.
It's amazing to think about all the potential applications of this technology. It feels like we're on the verge of a golden age of drug discovery.
I agree with all the advancements we've discussed, from AI-powered drug design to CRISPR-based gene editing and now cryo-EM. We're developing tools that will allow us to create personalized therapies that are more effective and less likely to cause side effects.
This deep dive has been truly mind-blowing. It's given me a glimpse into a future of medicine that is both exciting and a little bit daunting.
I know what you mean. It's awe-inspiring to think about what we might be able to achieve, but it's also important to consider the ethical and societal implications of these powerful new technologies.
As we wrap up this episode, do you have any final thoughts for our listener about the future of drug discovery?
I think the most important thing is to stay curious, engaged, and informed. The world of science is constantly evolving, and it's up to all of us to be active participants in shaping the future of medicine. The discoveries we discussed today are just the beginning of what's possible, and I can't wait to see what the future holds.
Well said. Thank you for taking us on this incredible journey through the future of drug discovery.
It's been my pleasure.
And that concludes our deep dive into the fascinating world of modern drug discovery techniques. Until next time. keep exploring, keep questioning, and keep learning.